fourth step, that is the transport of this active fatty acid to the matrix. So, on the inner side of outer mitochondrial membrane, listen to me very carefully, that the fatty acyl synthetase was present on outer side of outer mitochondrial membrane, but on the inner side of outer mitochondrial membrane there is another enzyme which we name as CPT1, like this. This is CPT1. CPT stands for carnitine palmitoyl transferase. It is enzyme of carnitine. What is a carnitine? Carnitine is a beta hydroxy gamma ammonium butyrate. The full name of carnitine is I am writing it over here, that the full name is beta hydroxy gamma ammonium butyrate. It's the full name of carnitine, and the carnitine is very important. We will discuss it in detail in another lecture. And here the CPT1, which is an enzyme of carnitine, and it is its full form is carnitine palmitoyl transferase, and its former name is CAT, carnitine acyl transferase. Carnitine palmitoyl transferase means it is for palmitic acid, which is a 16 carbon fatty acid. Now remember one thing that is very important for you, that this transport process or this carnitine shuttle is only used for the very long chain fatty acids, like the fatty acids which have a carbon length of more than 14, or in other words you can say that this is for very long chain fatty acids, and very long chain fatty acids their length is more than 14 carbons, So such type of the fatty acids are named as very long chain fatty acids, and less than the fatty acids containing less than 14 carbons, those are referred as medium chain fatty acids and short chain fatty acids, and this shuttle is not used for medium chain or short chain fatty acids. This shuttle system is only used for very long chain fatty acids after the activation. This is only used for very long chain fatty acids. So remember this point. And with the help of CPT1, this fatty acyl CoA, from this fatty acyl CoA is removed, and fatty acyl CoA is converted into fatty acyl carnitine. Here we have carnitine, and when this fatty acyl CoA enters into the space, here this carnitine will become attached to the fatty acyl CoA, and this fatty acyl group will be removed and it will become attached to this carnitine, and there will be the formation of fatty acyl carnitine. And CoA will be removed. Here you can write it as here. CoA is removed. Now once again listen to me, that first there will be the activation of fatty acid on the expenditure of one molecule of ATP and its two phosphate bonds, and with CoA, and the enzyme involved is fatty acyl synthetase enzyme, and fatty acid will be converted into fatty acyl CoA. This fatty acyl CoA, if it is very long chain fatty acid, it will need a carnitine shuttle, and carnitine shuttle, for the carnitine shuttle on the inner side of outer mitochondrial membrane there is an enzyme system which is named as CPT1, carnitine palmitoyl transferase 1, which will transport this fatty acyl CoA into the space, and in this space there is present carnitine. This carnitine, what will it do? simply it will become attached, or it will replace the fatty acyl portion from the CoA. CoA is removed and fatty acyl CoA will become attached to this carnitine, and it will result in the formation of fatty acyl carnitine complex. Now this fatty acyl carnitine complex, it will go to the inner mitochondrial membrane. It has access to the inner mitochondrial membrane, but fatty acyl CoA, it does not have access to the inner mitochondrial membrane. So that's why it has to be converted into the fatty acyl carnitine portion. Is that clear to you? Now from here there are two enzymes. Number one is translocase and number two is CPT2. This is CPT2. With the help of CPT2, first fatty acyl carnitine will enter into the matrix, and in the matrix this carnitine is removed and CoA is added, like this in the diagram you can see. And translocase is helping its transfer to the matrix, like this. CoA will be added, and as a result it will help the cell to again reform the fatty acyl CoA and plus carnitine. Once again listen to me, that fatty acyl carnitine with the help of translocase and CPT2, there are two processes. One process is with the help of this CPT2, and second process is with the help of another enzyme which we name as translocase enzyme, What will happen that when this fatty acyl carnitine with this help of CPT2 will enter into the matrix, carnitine will be removed and CoA will be added. When the CoA is added to this fatty acyl carnitine portion, there will be the formation of fatty acyl CoA and carnitine is removed. From here the carnitine is removed, and with the fatty acyl portion the CoA is added, and it will yield the formation of fatty acyl CoA, or it will be responsible for the formation of fatty acyl CoA. And what about this carnitine? This carnitine is shuttled back into the space, and now this carnitine is again available to form another complex, which is fatty acyl carnitine complex. And again the process will repeat, and again there will be the formation, there will be again the transport of one more fatty acid in its active form in the mitochondrial matrix. So listen to me very carefully once again, that first of all there will be the activation of fatty acyl CoA, then this fatty acyl CoA cannot cross the inner mitochondrial membrane. So with the help of CPT1, fatty acyl CoA is converted into fatty acyl carnitine, in which carnitine is added to the fatty acyl portion and CoA is removed. Second, this fatty acyl carnitine will get entry into the matrix with the help of CPT2, two enzymes, CPT2 and translocase. With the help of CPT2, it will enter into the matrix, but as it will enter into the matrix, CoA portion will be added to fatty acyl carnitine and carnitine will be removed, as you can see. This carnitine will go back to the space with the help of translocase enzyme. Now it is again available for the formation of another fatty acyl carnitine complex. So this was the fourth step, and this shuttle system of carnitine is named as carnitine shuttle system, and this is the fourth step of oxidation of fatty acid.